screen?
Yes, we can see it. Yes, that's cool.
This. Okay.
Yeah, perfect.
All right. So this is like a series of 2 talks. The first one I was asked to provide some introduction to methodologies, which I'll be using in my colloquium talks. So title of my talk is Stochastic Modeling of Reaction Diffusion Processes, which is a very big area. The goal of this 25-minute talk is just to introduce mathematical and computational methods for spatio-temporal modeling of biological processes, which will be used in my colloquium lectures. It's kind of designed for people who kind of are new to this area. There's not much time to go over everything in 25 minutes, so it's kind of like giving you an idea what this area is about. If you are interested in more details, then you can find it in this recent book, which he wrote with John Chapman, Stochastic Modeling of Reaction Diffusion Processes. This book is used in my institute as lecture notes for a different number of courses, including basic undergraduate level courses, also advanced graduate level courses. Over last year during the pandemic, we had to do everything online here, so I put some lectures also on YouTube. So if you are interested in more details, so you can just go to this link and find links to YouTube lectures, but also there are all computer codes, because it's a very computational course, which also introduces students to some computations. So I'll just kind of give a brief idea to the philosophy of the stochastic reaction diffusion models, and especially focusing on those which will be in my colloquium lecture. And I'll start with a very basic one here. So the basic one is just Brownian motion. So I can put references, which if you had that book, you can find it there. So this is just a typical model of Brownian motion, or the basic model of Brownian motion, when I have just a protein diffusing protein, which is, you can see in this video, and I'm calculating its position at time t plus delta t from position at time t. So it's kind of evolution rule. When I take my current position at some random displacement, this displacement is scaled with the diffusion constant, which is kind of physical property, but also with the time step of my simulations, how much I advance the time. And I just adding this normally distributed random number. So this is a stochastic model. I need a random number generator to do it. And if I do multiple realization of this model, I can just start multiple copies of this process. Then obviously each of these particles, given that they start from the origin, are given have different time evolution. So this will be a kind of model, stochastic model of diffusion. Such models are used widely for modeling intracellular processes, but my talk will be about multi-resolution modeling. So I'll be also looking at models written with different levels of detail, models which are coarser, quicker, but providing less detail, and models which are more detailed. So such a stochastic model can give rise to some predictable deterministic behavior, provided that we observe many particles. Obviously, at the final position of these proteins are not much of interest. It just depends on the random number generators. But what is of interest is what is the probability that the particle is a certain distance from its initial condition at some time t, or if I can provide some predictable deterministic modeling if I simulate many particles. So how can I do it? I can just take my domain and divide it into some compartments into mesh, and I'll be asking the questions: how many of these diffusing particles end up in each of these boxes at a given time? So then, if I simulate, say, a few millions of these particles, I obtain very deterministic profiles for probabilities of concentration. So here I simulate a few millions of particles, all started from the origin, and in these boxes there's about 1% chance that there'll be a final position after a certain time, and I'm just getting some Gaussian profile here. Now this can be mathematically described by partial differential equation. So in this case it will be just a diffusion equation, heat equation for the spatio-temporal density of particles. Now this was kind of like going from a stochastic models to description, which in terms of some kind of predictive deterministic quantities like densities or probabilities, getting deterministic model. But I can also go the other way, ask what is the underlying more detailed model for stochastic Brownian motion. So that's kind of like again describing this in this course later, because Brownian motion of a protein large particle is a result of collisions with many small particles. So I can use just Newton's laws of motion to simulate it. And in my talk, I'll be discussing molecular dynamics models, which are kind of slightly more complicated version of this caricature model. So what you can see in this video, you can see big particle, just a ball, which is colliding with tiny particles, which are caricatures of water molecules. All these particles, you can go see that all of them just go straight, all the blue ones, until they collide with the big particle. But because there are many of them, so that although the evolution is kind of deterministic, because there are many of them and they're kind of have random initial condition, then it provides this stochastic trajectory. So you can see when I'm just plotting the trajectory of this particle, is doing exactly the like a similar process, similar Brownian motion as I had before. What is the take-home message here is that this always a deterministic models. After I provide initial condition, everything is purely deterministic. Everything just goes straight, and they collide. Collisions are provided using conservation of momentum and energy. It can appear quite stochastic, and can be also possibly describe some stochastic process. So what I told you on this like an illustrative example so far is that for Brownian motion, I can have a very detailed deterministic model where I simulate not just the proteins, but also surrounding medium, surrounding water molecules. I can then have a much coarser model when I kind of remove the waters and describe everything in terms of stochastic model just for these important molecules, say proteins, which are diffusing. Or I can have a coarser model if I have many of these, I can ask in the density profiles. So for every application, we can create such a sequence of models. We're going from the most detailed to the very coarse model. And if we decrease the level of detail, then it leads to also the decrease of the computational cost. So this is difficult to simulate, or it's computationally intensive to simulate, because I have to track all the particles, not just the proteins, but also all the water particles. Here I'm simulating just partial differential equations, so I don't need to worry about individual positions of these black particles. And for each application, you have to choose a model which includes as many details as necessary. In my colloquium talk, I'll be discussing multi-resolution modeling, which is about using models with different level of details in different parts of the computational domain. So I'll be trying to merge these different descriptions in the same simulation. Now, I think in the rest of this introductory talk, I will kind of talk a little more detail about these stochastic models for reaction diffusion processes, because there are many of them. And there are several different approaches to stochastic modeling of reaction diffusion processes. What I described so far is just model of diffusion, one possible model of diffusion at this level, when I just simulate individual particles. So going then forward, discussing reaction diffusion modeling, I just decided to put here one illustrative example. So from the point of view of deterministic modeling, it's everything modeling in terms of partial differential equations for concentrations. So this will be a PD, which I already showed, the diffusion PD. But now if I also have some interesting processes happening, some reactions, there will be also some reaction term. So this here CI, the concentration of the I chemical species. I have a combination of chemical species, which is each of them diffusing, and reacts according to some reaction term, according to the underlying biochemistry. And such equations can lead to patterns. Just going to put some like examples of pattern. The patterning problem which I chose for this talk is the kind of, I would say, the simplest one. It's about a morphogen gradient. So my model is just that I have diffusing morphogen, which is produced in the left part of the domain. So it's produced somewhere here. It's produced, and it's then diffuses, and it's also degraded everywhere. So as time progresses, I start with no concentration in the of this chemical, of this morphogen in the system, and its concentration is increasing and creates this gradient. Now, such a model see as a kind of basic caricature model in developmental biology for creating patterns. Sometimes called French flag model, in a way that you can assume there's a underlying layer of cells, and these cells can interpret different levels of the signal. So if they see a lot of concentration of this morphogene, then they start a blue program. If they see intermediate concentration, then they start a white program. And if they see a little concentration, they start a red program. So there are some thresholds which they can interpret, and they can kind of read the information about the chemical, about the morphogene in their environment, and use it to determine positional information. So in this way, if there is some little pre-patterning, that there are a few cells here which can secrete some morphogene gradient, then this can lead to more detailed patterning. And that's called French flag model, because just the colors are chosen here according to French flag. So it's kind of a caricature model, and you can use then take this layer of cells to get some patterning. Now, what stochastic reaction diffusion modeling does for such patterning system is just to try to add more details, to use stochasticity level of noise, that there are not just infinitely many copies of these morphogenes, but only finite copies. So what I'll be discussing now is just that how to model it stochastically. So and you can see that if I use a stochastic model, which is just describing everything in terms of number of molecules in the domain, then I'm getting a noisy pattern. This is just one realization of stochastic reaction diffusion system. Again, I kept the same thresholds. You can see that there is some underlying pattern, but also there's some fluctuations at these boundaries. So that's what one tried to get through stochastic reaction diffusion modeling, and how to actually model this in the computer. So there are different ways how to do this. I'll just highlight kind of two main approaches. If you are more interested in modeling biological systems, so all these approaches have been coded in different software packages. So I can find many of them online. Yet some of them are better supported than the others, but they are available in the literature. What I can just showing you these snapshots of various websites that I could subdivide them into two main categories. Some of them use lattice-based simulations, like some called compartment-based reaction diffusion modeling, and some of them they track individual molecules. They use molecule-based approaches, some of these software packages. So I'll just on this example, on this like a morphogene gradient, explain what is actually main differences between these.
So Radek, what do you think? What is the best for you among these many packages? Can you recommend the best one for the people?
Well, I work with some of these people, so I could say we contributed to develop a small dyne, which is a molecule-based model. Sort of this is from Zan Schulten group. Also we collaborated. So this one kind of has like actually they all are supposed to was parallel computing to simulation on GPUs, because at the end you end up with very big systems. So you need for each of them you always check, is it supported? Are there every year new releases? And if yes, then this is the software to invest your time in. Because you will get something which is tailored to the most modern software architectures. Some of them, like you can just look that they were maybe developed by corresponding developers, but then they perhaps lost funding or interest, so that there is no new release. So, but there are new being released, but they would always decide which of these products look more long term and supported. But what I would like to say in this rest of this, in the rest of my talk, is what is actually underlying methodologies behind them, what is compartment-based modeling and what is molecule-based approaches. So let's just illustrate it on this model. So I had this morphogene gradient. There was a production of molecules in part of the domain and degradation in the whole domain. So the molecules also diffuse, and the initial condition is there are no molecules. So that was the system which I showed you in my simulation. And if I want to do it compartment-based, I take my domain. So this is kind of a elongated pseudo one-dimensional domain. So I just divide it into compartments, and I describe the system in terms of numbers of molecules in each compartment. So the underlying variables will be, if I have here 40 compartments, there will be just 40 numbers, where I is the number of molecules in the I compartment, and diffusion is then more or less jump process between these compartments. So I can write it in chemical reaction formalism. So it's actually just, if I have one chemical, suddenly I have capital K chemicals, or 40 chemicals, if K is 40. Suddenly a chemical system can grow from relatively small chemical system to very large chemical system, but it's still a chemical system, and there are efficient algorithms for simulating such chemical systems. So this is kind of a diffusive jumps, simulating jumps between compartments, the first, second, and then there is a degradation everywhere. So everywhere there's some destruction reaction, and the first in the red part of the domain is a production reaction. So this will be kind of underlying concept of this compartment-based modeling. Let's just not track individual molecules, but only track numbers of molecules in each compartment. For molecule-based modeling, if I want to simulate the same process, I don't have any compartments. So I will have to track individual molecules. So in this case, I have to simulate trajectory of every molecule using the Brownian motion. So this is kind of the equation which I started with this brief talk. And then I have to just think how to introduce reactions. So in this case, it's just that every molecule can be destroyed in the simulation. That would model degradation. They can diffuse, their positions are updated by this rule, and then also the molecules are produced randomly to the system. If I have a more complicated system, then I have to start thinking how to do bimolecular reactions when two molecules meet and react. So there's a lot of estimations of distances between molecules, and molecules react with some probability when they are sufficiently close to each other. So this is kind of underlying difference between these two types of approaches to reaction diffusion modeling. Now, if I want to apply them to this particular model, so that would be kind of it's kind of as a figure taken from the textbook. So we can see here the video which already saw the difference from this video and the first video was that I just, in the previous one, I put some biological interpretation. So I used through some thresholds and I color it to get that French flag model. But what I do here, I simulate individual molecules. So you can see there'll be a lot of individual molecules to simulate, but at the end, what I visualize is just number of molecules in some bins. So I divide my domain into 40 bins and plot the number of molecules in each bin. So while I'm visualizing 40 numbers here, to get this number, I'm simulating 100 individual molecules. So I'm simulating several thousands of molecules towards the end of simulations, tens of thousands, but I'm only visualizing few numbers. So one can just think that this could be, if I'm only interested in this type of gradient formation information inefficient, because I'm simulating a lot of information where each individual molecule is, but I throw it away. So that what compartment-based modeling doesn't do it. So that would be what I have here. Simulation looks the same, yeah, but what is happening in compartment-based modeling? I only simulate 40 numbers, so it's more efficient. I'm using something called Gillespie algorithm, or it's equivalent formulations which are more efficient. What I'm comparing it with solution of deterministic model, which in this case will be a reaction diffusion partial differential equation that I have a diffusion in one dimension. A is the concentration. They have some localized production in this part of the domain and degradation everywhere. So this would be kind of like a conceptual difference between these models. In these simulations, I can get the same pictures using compartment-based models and molecular-based modeling. So there is not much need to use molecular-based modeling, which will be more computational intensive, but there are some other more complicated examples where it is needed. So I think if I kind of try to summarize this like an introductory talk, so I introduce these different approaches. I introduce very detailed molecular dynamics type modeling when I simulate molecules and their environment. But I also discuss the stochastic reaction diffusion modeling. The most coarser of these approaches was compartment-based modeling, when I divide the domain of interest into small compartments. In my example, this was one-dimensional, but we usually do it like in this lattice microbes. That's a simulation three-dimensional space of the whole cell, of a significant part. The diffusion is that model as jumps between these compartments. and chemical reactions are localized and can only happen in molecules between in the same compartment. So small compartments are effectively assumed well mixed, but by having a lot of them, you can simulate spatio-temporal distribution. So such a modeling leads to a large system of chemical reactions, which is in itself how be efficient to simulate such large systems. But that can be done. Some standard algorithms, a Gillespie algorithm, but there are some equivalent and more efficient formulations which can be done, which are implemented in these software packages, implemented also in the parallel way simulations of graphic processing in GPUs. That's kind of very popular to speed up these simulations. Now, I also discuss molecule-based modeling when we simulate trajectories of individual molecules, which is more detailed, provides more detail, but it's also more computational intensive. So we have to choose it only when it's needed. So for my examples, was, for example, not needed because I could get the same information from both. But once we start to simulate bimolecular reactions, then interactions of particles, then it's very useful and it's implemented in small software package SmallDyn. So my colloquium lecture will be on multi-resolution modeling. So I'll be discussing these models and also these very detailed molecular dynamics models, and be asking the question how we can use these models with different level of details in different parts of the computational domain. So if you were kind of, if this was too quick for you, then I just kind of mention it. All these like slides were taken from various courses, and then this courses takes off over they can over 20 hours. So I kind of like took slides and videos from very different parts of these courses. So if you saw it for the first time, then please don't be discouraged. As I said, videos provided on YouTube, and there's also these textbooks where you can find more details about what is behind it. I kind of use this to introduce methodologies, which I'll be discussing in my colloquium lecture. So this could give you some idea what my colloquium lecture will be about. So I think that's all I wanted to say.
Cool. So Radek, thanks so much. We have some couple minutes for the question from audiences. Any question? Please unmute yourself. You can ask a question. Yes.
Thank you, Radek. That's very interesting. So in the last slide, you showed some probability distribution fluctuating at each bin, and you showed also deterministic PDE. So is the PDE the average of the system at each bin, or does it mean something else?
So it's a very good question. So maybe I should just
here again. Yeah.
Share the slides. Maybe I will share the slide again.
Oh yeah, thanks.
So I'm sharing.
Here we go. Yeah.
My slides again. Yeah. So the question was about these pictures when I showed the molecular-based model or the compartment-based model, and I was producing this black line. So I actually put in my pictures that it's actually a mean. So it's an average.
Yeah, there we go.
In this average describable, but this is specific for this model. That mean is it can be estimated. So if I want to calculate averages, I have to calculate not just one realization, which was showed in video. I have to calculate at least thousands of realizations. So it's additional computational intensity. But then I can estimate average number of molecules in each of these bins, and I can obtain these black lines. So that's what is then in this specific model. One can actually prove, because it's just first order reactions. So one can prove that it's described by this partial differential equation. Or obviously, when compartment-based model, that proof is going that it's described by something called reaction diffusion master equation. And if the bin size goes to 0, then it converges to this partial differential equation. So that's going to be proof. And the molecular-based modeling, there is already no bins, so that there is diffusion is described by this process. So that's, if I simulate it, it's exactly described by this equation in terms of like a probability densities. But if I want to calculate it, I again have to discretize this operator somehow and plot it. So there is some underlying discretization, which is kind of the size of this bin in both of them. Here it is used for evolution of stochastic model, because I'm using compartment-based model, compartment size, to actually calculate the stochastic trajectories. Here I'm only using this discretization to visualize my underlying molecule-based model, because there is no compartment. But if I want to calculate the solution of the discrete solution of this equation, then I will have to also discretize it, so with the corresponding step size.
Okay.
But in this case, it's the PD describes the mean. It's not true for bimolecular reaction. If there was some bimolecular reaction, one can prove that it's not the case.
Okay. There is a question from Selik. Selik, you can unmute and then you can ask a question.
Hello, everyone. Thank you, Professor Erban, for this interesting talk. My question is about your compartment-based reaction system, and if I have seen correctly, the reaction rates were constant, the same constant. All of them were D's, some D's. Does this really happen in a biological system? This is my first question. And the second one is, what if we have different reaction rates occurring different speed? How this would be looking like?
That's a very good question. So kind of so I'm simulating the diffusion process here with some diffusion constant D. So I'm thinking for this basic model, which I kind of have here, there is some underlying physical quantity, diffusion constant. So I assume it's kind of homogeneous. The diffusion constant is same. Now there's this lowercase d, which is just a diffusion constant divided by compartment size square. So that's kind of how one realizes this lattice-based or compartment-based model with this model. So this capital D is this lowercase d multiplied by compartment size square. Now, if I have processes with diffusion constant, which is space dependent, and that would kind of reflect that there will be a different D's. That's again possible and used in some applications. Again, I took this from slides from that course, and that course it follows by another slide when these D's are actually different. So it leads to not to reaction diffusion systems, but also to reaction diffusion advection systems. So there is also some underlying advection term, and it's useful for modeling system, and there is some active transport. So this was more applications when I'm considering diffusing proteins, but if these particles were not molecules, by say diffusing bacteria, and then they are not just diffusing, they have plenty of active transport, they respond to extracellular cues. And therefore, these rates would be dependent on extracellular gradients. So that's is definitely a very good point. And yeah, this was just a simplest model to introduce this like a conceptually compartment-based modeling and molecule-based modeling. But it can be implemented in both type of approaches, and again has been in the literature. That these constants can be varying in space, as well as this diffusion constant could vary in space.
Okay.
Thank you very much. No.
Great. I think.
May I ask one?
Oh yeah, you can. Quick question, because we don't have.
Yes.
Yeah.
This is going to be a quick question, I think. And I have a question about this comparison of this compartment-based model and molecular-based simulation. So in this system, there are degradation and generation of the particles, right? How do you model this in the molecular-based side? I mean, is the particle going to disappear at some point with some probability?
Yeah, it's a good question. I didn't put it here, but so each particle will be similar. This position by this rule, the position at next time step is position in previous time step plus some random number, which is normally distributed corresponding to scale. But then you have variable number of particles. So in these videos, you have to kind of be introduced, and you have no particles at the beginning, and you have to keep some data structure, which is adding particles according to some rate, which is consistent also with this production rate here. So to make it exactly the same, the production rate per compartment per unit of volume has to be the same. So you generate some Poisson random numbers to produce the particles, or exponentially random numbers to produce them according to this rate. And on the same, each particle has some like a mean half time, so they're degrading with this rate, so each particle can disappear from the system.
I see. Thank you.
Cool. Okay. So maybe you can ask the more question after the colloquium. So okay. So maybe Radek, could you share your colloquium slide now?
Okay, I'm trying to share the screen.
Yes.
Am I sharing the screen?
Yes, it's good. And then maybe you can make a presenter mode. Because there are new people came, so let me introduce one more time Radek Erban. So I have the great pleasure of introducing Professor Radek Erban as today's BIMAC colloquium speaker. In a series of landmark studies, Dr. Erban has applied the theory of stochastic dynamical system, in particular, the stochastic reaction diffusion system, to problem in various biological problems such as intracellular dynamics of molecules. Dr. Erban is a currently Royal Society University Research Fellow and a professor of the mathematics at the University of Oxford. He is also editor of the 5 distinguished journals, including the Journal of Mathematical Biology and European Journal of the Applied Mathematics. The topic of his talk today is multi-resolution methods for modeling intracellular processes. Please join me in welcoming Dr. Erban.
Thank you very much. Thank you for this kind introduction.
Mm-hmm.
So this is kind of like a continuation of talk, but my understanding is some people will join just for this talk and some people were already on this like a tutorial at the beginning. Mm-hmm. So I'll kind of mention maybe perhaps with the most important bits from these methodologies which I was discussing before to try to make it like independent. So I'll be talking about multi-resolution methods for modeling intracellular processes. So my talk started with just with a textbook picture of an eukaryotic cell. So it's just this is just taken from a textbook, just to highlight that currently we have a lot of information about intracellular processes. We know a lot of details, and if we zoom any particular region of the cell, we see any more information. Mm-hmm. The question is, if you want to put such information into a computational model of the whole cell, mm-hmm. Then this is kind of a hopeless task, mm-hmm. because it's impossible to use all the available information which we have about microscopic level, about structure of all the proteins or all the biological processes to create a computational model of the whole cell dynamics. So in some applications, which are discussing microscopic details only required in a relatively small region, for example, close to a cell membrane or particle organelle. Mm-hmm. So in this application, what is useful to do is something called multi-resolution modeling, which is using models for different levels of detail in different parts of the computational domain. So that's what we might talk about, telling you about the underlying mathematics, but also about some applications where we use such methods. Now, to kind of help you to orient in my talk, I kind of highlighted here the main methodologies which I'll be talking about. Mm-hmm. So I will kind of focus on 2 types of multi-resolution methods. I'll be discussing effectively these arrows. So in the most detailed modeling methodology which we use is our molecular dynamics. It's a classical molecular dynamics, starting from all atom molecular dynamics simulations. But some models we built also with a little Brownian dynamics, and we just do stochastic modeling of individual particles. Mm-hmm. Those we were on my previous talk, that was what I was calling the molecule-based modeling of stochastic reaction diffusion modeling, and there are much coarser models which are compartment-based models, which can be analyzed mathematically using reaction diffusion master equation. So I will discuss these arrows, how we can go from more detailed model to coarser model, and how we can use these limiting processes to design some efficient computational schemes to answer the questions of interest. So I'll start with this red arrow, which is kind of our success story, which is a joint work with many people. I could call it a success story because in this area we have papers not only on the development of mathematical methods and their analysis, so we know how accurate these methods are, but also on applications, and there's also methods were implemented in some of this computational biology software, which I showed in previous tutorial. The blue arrow is just a work in progress, something which interests me a lot, and when I have time, I'm also thinking the blue arrow, we publish some papers as well. Some of them are published just by me, like a single author papers, but I also have some collaborators kind of in this area. So I'll start with the red arrow, and I'll just, since the Brownian dynamics is in the middle, so I'll just kind of first tell you what I mean under this general title of Brownian dynamics. So in the simplest possible setting, Brownian dynamics will be just a discretized Brownian motion. So I have a protein here, and I'm asking about its time evolution. So I'm asking what is the position of this protein at time t plus delta t, given my current position, and I can calculate it by adding some random normally distributed displacement to it, which is scaled with diffusion constant and with the time step of simulation. So if I use such an evolution rule multiple times, I get effectively the textbook picture of a Brownian motion, which is shown here, and which I showed some videos in my tutorial. Now, mathematically, it's just a discretized stochastic differential equation for the Brownian dynamics. So this will be what could be considered as an underlying model for moving of particles. I'm studying individual trajectories of all proteins or important molecules in the system. Now I have to add reactions to it. So the reactions can be of various forms, or interactions between these particles. The most challenging are bimolecular reactions, or reactions when two molecules are interacting. So I have to kind of, they are simulated often like that. I simulate trajectories of individual molecules. I'm checking in my computer code distances between these molecules, and if they are sufficiently closer than some reaction radius, then these molecules can interact with some probability. So this is kind of underlying way how to treat bimolecular reactions, but obviously there are also first order processes like a degradation reaction, which kind of each reaction can degrade, which has been implemented in a number of software packages. I put small dian in which this multi-resolution modeling, which I'll be discussing, is implemented, which is a Brownian dynamics, started as a Brownian dynamics software package, but then now there are, like I said, there's a support how to coarse-grain parts of the domain to compartment-based modeling using some of these results, which I'll discuss here. So this is kind of a kind of quick introduction to what is Brownian dynamics. And
so Radek, the binding probability approach and reaction radius approach lead to the same result, or are they different?
I didn't hear it much.
My question is, I guess reaction radius approach is when two molecules are under a certain radius, the reaction occurs, right?
Yeah, that's kind of like an underlying like a method to implement reaction, that if there's some reaction radius, and if the molecules are sufficiently closer than that in my, then some interaction could happen with some probability.
Some probability. Oh, so this is not to distinguish the approach, just the same approach.
It's like the same approach. Yeah, it could happen with probability 1 in some codes. It's basically the molecules just react within certain distance. In some of them, they need to take into account that not every collision is effectively reactive collision.
Okay.
So,
Got it.
That it react with some probability. So there's it's a conceptually, similar like an approach. They react if they are close with some probability, which could be 1 in some of these codes.
Okay, thank you.
So this is kind of Brownian dynamics. I've got compartment-based modeling, kind of introduce it in my tutorial on some like a one-dimensional model. It's effectively, I'm not simulating individual molecules, but I'm take my domain, divide into compartments. So in this like a domain, I divide it in it's a 2-dimensional domain. So into some squares. In real simulation, it will be in cubes, and I kind of describe a system by number of molecules between in each compartment. So if I was thinking about individual molecules, then the process will be effectively some lattice-based random walk. So jumping between compartments with rate d over h squared. So d is my diffusion constant, h is the compartment size. And if I consider a random walk when this molecule can jump with this rate d over h squared, and then I obtain diffusion process which with the same diffusion constant. But the big advantage of compartment is I don't simulate individual molecules, so that I'm not simulating individual molecules, but I simulate the system in terms of numbers of molecules in each compartment. So in this state, there are 7 molecules, here's 8 molecules, 4 molecules. I only evolve these numbers. I don't evolve individual molecules. So we do not compute trajectories. So we only simulate the time evolution of numbers of molecules in each compartment, and mathematically leads to a discrete space continuous time Markov chain, which can be simulated in a similar way like the Gillespie algorithm simulates a well-mixed system of chemical reactions, which makes the compartment-based modeling more efficient in a way that it has been used to various modeling of whole cell processes. I can put here from some of these packages. So this is from lattice microbes, simulating the whole cell modeling as early as 2014, 2016. It's a parallel computation, so it has to be on GPUs. So this is kind of like a quicker way how to stochastically model reaction diffusion processes.
So Radek, there is a question in the chatting room. Is it enough to consider only horizontal and vertical movements except diagonal movements?
Oh, it's a good question. Yeah. So in this, so this is kind of like a 2-dimensional, and in this would be a 3-dimensional, so be a jump in 6 direction. Mm-hmm. Yes, it's enough because you do diagonal movement. You can just go like this. So what you want to achieve with this lattice-based model is that if it's purely diffusion, you would like the same statistics of the diffusion process like the individual based model, like at the level of coarse graining. So, if I simulate only diffusion, if I didn't introduce reactions, then the probabilities of being in these compartments will be the same if I simulated individual molecules. What makes it a little different is that once I start to simulate reactions, but for diffusive jumps, I don't need to worry about the diagonal.
Okay, wow, okay. Thanks.
So this is kind of like a quick introduction of what are these methods and what are the advantages or disadvantages. And now I'm going to be discussing multi-resolution modeling, which is about using models with different levels of detail in different parts of the computational domain. So this is kind of an area which is very much of interest to mathematicians like me because they are well-defined mathematical problems, but also it's useful in applications. So what I mean by well-defined mathematical problems is that I just wrote it as like a caricature of a domain, and I have in half of this domain I'm using compartment-based models. So I basically just track numbers of molecules in the compartments, and in the other part I'll be simulating individual molecules. And I want to kind of design the boundary condition, how to kind of transfer molecules from left to right and back in a way that in this part of the domain, when I'm using the most detailed model, I'm getting the same results as if I would get them if I was using detailed model everywhere. So effectively, this is kind of a part of the domain where I'm not so much interested in the individual molecules, but I would like to have the same statistics, same results in just the smaller parts of the domain. So when you think about it like in this, you might just start thinking, well, this is kind of conceptually quite easy. For example, for this connecting Brownian dynamics compartment-based model, because I could just say, well, if I'm thinking about the level of individual molecules, I told you that molecules are just doing some jump process on a lattice here with some rate constants, which is diffusion constant divided by compartment size square. So I could just let it jump. And when it's in this compartment, then there's just some jump rates to all other direction, but also a jump rate out. So I could just generate some random numbers, and I could just, with the same rate, move these molecules here, then kind of have to initialize it. It's more detailed model, I have to tell where exactly it is, but I could just say maybe it's at the middle of the summer, like a pseudo compartment here. So I would just put a distance, which is half compartment size, and simulate it as a Brownian motion. And if this molecule wanders to this compartment, if there were 7 molecules, I could just change it to 8. So I could just basically glue these processes very easily and have a multi-resolution model. What is both interesting from a mathematical point of view, interesting from researchers in the era that if you do it in this way, that you can obtain very incorrect results. In a way that if I just simulated pure diffusion, so I have a diffusion of particles, so I'm simulating here only diffusion, there are no reactions, and I'm just doing this basic process that I'm transferring molecules according to corresponding rates between this artificial interface. Then I can just ask what is happening at equilibrium, how many molecules are on the left and how many molecules are on right. And a typical result which you would obtain if you just glue two computer codes together would look like this. So this is kind of position. This is when I simulate a compartment-based model. This is where I simulate compartment-based model. This is where I simulate molecule-based model. So here I'm simulating just the boxes, number of molecules. Here I'm simulating individual particles. And if it's pure diffusion, what I would like to get, I would just like to get that is a constant profile of molecules. So there's basically, if I was simulating two diffusive processes, the equilibrium profile would be just a constant. You can see that it's indeed the case. If I look in this part of the domain, it's a constant, it's a diffusion, so it's a homogeneous, it's here. But what I'm going very wrongly is how many particles are actually on the right or on the left. It looks that there is some like a boundary, something which is not correctly implemented. It looks that I'm getting more particles are jumping more likely to the left than returning back to the right. And there's also some like a destruction of this close to the boundary. So this was kind of like it's like a naive coupling when I just let run in parallel two computer codes and let molecules pass according to the corresponding rates. So if you want to try to explain why this behavior, which is obviously undesirable, happens, then you have to kind of look in more details. What is the underlying mathematics? So let me just explain why this can happen if you just glue two computer codes together without doing anything in addition to it. So that's what I kind of like showing on this picture. So this is my undesirable results. I would like just a constant homogeneous profile for the diffusion of molecules. And this is kind of in my simulation, I have this interface, this artificial interface, and I'm simulating Brownian motion. So on the right, I'm simulating individual particles according to this evolution rule. So I'm simulating the position at time t plus delta t for position of time t by adding some random displacement. So this is my time axis, this is time t, this is time t plus delta t. This is my molecule-based model, this is my position at time t, and this is position at time t plus delta t, but delta t is my time step. So you can see if my simulation, if I simulate it over one time step, I know that I'm in the molecule-based domain, I'm in this part of the domain, and at the end I'm again there. So if I simulate it with this time step delta t, I have no reason to be transferring molecule from right to left. But this is the molecules are not going straight between these. If I refine the time step, if my time step is much smaller, 100 times smaller, then I don't get this straight trajectory. I get, for some trajectories could go like this proper trajectory, some trajectories could go like this green or red trajectory. In particular, if between my simulated time steps, the molecule was following this red trajectory, then it actually crossed this interface, and I should be transferring it to my compartment-based simulation. Or if it went this one, that will not cross the interface. So the take-home message is that depending on the time step, which is internal parameter of the simulations, I can get different number of times crossing this boundary. If I use coarser time step, I didn't cross it at all. But if I refine the time step, there's some probability that molecule would cross the interface between the time step. This probability can be calculated for strain interfaces, and it's basically given by this number. So how it depends, basically, this is my position at the beginning of the time step, this is my position at the end of the time step. So if one of them is 0, so which is my boundary, then this exponential will be 1. So I'm definitely hit the boundary with probability 1. If I'm further, like here, with some distance, then this is just something smaller than 1. And if I'm very far, it's probability 0 if I was very far from the boundary. But if I'm kind of getting closer and closer to it, this probability is non-zero. And it means that I have to take into account that even between time step I hit the boundary, I have to kind of take into account that this hitting probability depends on internal parameters of the model. So the correct coupling must depend not just on the measurable quantities or physical quantities like diffusion constants, but also on the internal parameters of both molecule-based and compartment-based model. For example, on the time step in this case of the molecular-based model, which I'm using to evolve individual molecules in this part of the domain, or on the compartment size, when I'm doing these jumps between compartments, I kind of discretize it with some compartment size h. So they both have to be included, because as I change them, I kind of changing how the molecules, how frequently they are crossing the interface. So that's kind of like it brings the additional challenge, how to find these transition probabilities, which correctly just simulate this transition molecules. For this example, simple example, I would just like to get constant profile.
So Radek, I have a question. So if I understand correctly, so on the molecular-based domain, you discretize the time, and on the compartment-based domain, you discretize space.
Yes.
So we have to couple this discretization in some sense, right? To get the good simulation.
Yeah, so there are different like a discretization. There's obviously time on the left as well. I discretize the space, but time is evolved by Gillespie algorithm. So it's a randomly sampled according to some rates. So when these molecules transfer. But so there's no time step on the left, and there is no spatial step on the right.
That makes sense. In that way, the coupling should be depending on the delta T and H, right? Somehow.
Yeah.
Yeah. And then there is one more question in the chatting room from Kyunghan Che. Is the result robust in terms of number of the compartments? I mean, the result of the difference between the equilibriums for each division in space. In the chatting room, there is a question. Can you see?
I don't see the chatting room question.
If you click the chat in the bottom.
Actually.
Oh, you can. Yeah. Kyunghan, you can.
Yeah. I don't see the chat here.
Don't worry. Yeah.
In my opinion, if the number of compartments increases, then the equilibrium, the green region can be decreased. Yeah. So because there's no way to, there's no how can I say, sort of free direction to go out, but in the molecular-based model part, the particle can go anyway. So that's why I think the difference can happen in this simulation.
Yes, so you are right. It depends on the compartments. If I'm kind of when I'm going here, I was just saying the jumping rate is d over h squared. So if I have more compartments, if my h is smaller and smaller, this is h, my compartment size, then I'm jumping quicker between these compartments, because I have to jump quicker per unit of time to go across the whole domain at the same amount of time, roughly. So that's kind of so by introducing more and more compartments, I'll be jumping here quicker, and also that would also kind of affect how frequently I'm hitting the boundaries, because the jump rates from this compartment will be, if it's half compartment, boundary compartment, I can be jumping with rate which is depending on h. As I'm seeing here that the jumping from this one to the left depends also on the time step. Smaller the time step, more hits of the boundary. You're right.
Cool. Great. So indeed, if you have any question, you don't have to write in the chatting room. You can just unmute and then you can ask a question to Radek, right?
Yeah, that'd be great. Because I can't sharing what I don't see the chat.
Yes. Cool. Okay, thanks, Kyunghan. So go ahead, Radek. Yeah.
Okay, so then just basically start thinking. So what has to be done to make to remove this boundary layer which you saw there? So basically, you can approach it in different ways, but I told you that the jumping rate is d over h squared. D is diffusion constant, h is the compartment size. So in my numerical scheme, I'll say that the particles jump from the boundary compartment to this Brownian domain, not with the rate d over h squared, but with some rate d over h squared times phi. So I kind of modify the rate. It cannot be 1. It should be something else. We kind of agree that it should depend on time step, space step, and diffusion constant. So there'll be some and this phi is to be determined. Now, if particle jumps from this boundary compartment, again, I have to kind of maybe think about it where I will place it, because here the particles are just in the compartments, but here they have individual positions. So I have to initialize it, and how far I should initialize it. So I'll be doing according to some probability distribution. So this distance x will be sampled sometimes closer to the boundary, sometimes further boundary according to some probability distribution. And I could just say that all particles which interact with the interface, which kind of cross this boundary back, will just be adsorbed to that compartment. But what I did by this modified scheme, I introduced extra coupling parameters, this rate and this distribution. And then my mathematical question is how kind of related with the experimental data. In this case, it will be just diffusion constant, and also with compartment size and algorithm parameters, compartment size and time step. So to do it, one has to write some underlying equation for probability in this case. So compartment-based models is described by a master equation, reaction diffusion master equation, by probabilities being in compartments, while this is described by probability density functions. So I can just quickly show the equations which you'll be working here. So this is basically compartment. You can, those who kind of are in this area will kind of recognize that this is just a lattice-based random walk. This is basically just a compartment-based jumping from the boundary compartment. I can just jump from the neighboring compartments or jump out of it. But I can also come to the boundary compartment by diffusing from a continuous domain, and in the Brownian motion just describing by this continuous, there's some coupling. So I can write underlying equation. These equations have probabilities which will include these unknown quantities f and f, phi and f. And mathematical question is how to find them, that there is no boundary layer, so there is no error in the simulation. So this is done using methods of match asymptotic expansions. It's in these papers, but I'll just show the final results, which the equations look complicated, but the final results are not that complicated to implement. So if you do this analysis, the final result is that this rate is not 1. That we kind of observe already from our illustrated example, but it kind of depends on h and delta t using this formula. So it's basically depends on my compartment size and time steps. So depending whether how compartment size and time step are related, I could get either a rate which is greater than 1 or less than 1. So my picture, which showed that there's more molecules on the left, could be also reverted if I just chose slightly different h and delta t if phi was equal to 1. So 5 cannot be equal to 1. It's chosen by this formula, and f is just this error complementary error function distribution. So I'll just show you a movie how it kind of works when I implement these parameters, which kind of highlight all the points here. So what I'm showing you in this movie, this is the movie where I'm just showing results of 4 different simulations. So this is just a reaction diffusion system, traveling wave. So this blue line is a partial differential equation. So it's basically a solution by reaction diffusion PD, it's a traveling wave. And now what I'm showing here in red was a compartment-based modeling. Can I just run it again? So compartment-based modeling is in red, and molecular-based modeling is in blue. So you can see actually that if I use purely PD or molecular-based modeling on compartment-based modeling, which are these plots, I'm getting different traveling wave speeds. Yeah, PD is quicker than molecular-based modeling, but if you look at the bottom simulation, compartment-based modeling is actually quicker than PDE. What the top shows the results of multi-resolution simulation, when you can see that it has the same speed as molecular-based modeling, because that's what you want to achieve. This is your ground truth. This is the most detailed model, which are simulate individual particles which are reacting to some bimolecular reactions. So the speed is the same, but it's achieved by having only at the tip, when there is a low number of molecules, having molecular-based modeling, and behind it's use all the compartments. So this is kind of much more efficient than this simulation, but provides the same quantitative information at the level of molecular-based simulation. So that's kind of what is the highlight. So that works also not just for equilibrium simulation, but it's just for traveling wave, that I kind of replace all the molecules behind the front of the wave by compartments, and I get the same results as I would get from molecular-based simulation. So this is the computational intensity of the bottom simulation, but the accuracy of the molecular-based model. So it's kind of what implements these formulas. So if I kind of if I conclude this part of my talk, this was just about coupling Brownian dynamics with compartment-based modeling. So what I showed you kind of underlying issues that one has to do it a little bit care and to do some extra degrees of freedom, which they have to be carefully matched. There is, as I mentioned, some software implementation and small dyne. We just mention some applications as well. We apply it to different intracellular processes. I put here modeling calcium dynamics and actin dynamics. So just the next two slides show type of applications where we use these type of models. So one of them is the intracellular calcium dynamics. So this is intracellular calcium induced calcium release. So it's the calcium which is released from endoplasmatic reticulum through some channels, and these channels can open or close, and they are controlled whether they open close depending on individual ions. So there are some binding sites to which this individual ions can bind. So you simulate them close to the channels using Brownian dynamics. And when certain number of ions bind, then these channels are open and a lot of calciums are released inside the cell. So if you look how much concentration of the calcium is a function of time in this type of system, is that this time, this is concentration of calcium, there's almost no calcium. So in this type of simulation, you can easily do Brownian dynamics, molecular-based modeling. But once the channels open, there's such a huge influx of calcium that it's going to become computationally unfeasible to pass this peak. And because what happens then is the channels then close, and the calcium is pumped out, and there's again low concentration of calcium. So there's these kind of calcium spikes. Now, in terms of underlying simulations, there are some little pockets of simulations when you have the channels when you use Brownian dynamics, but the rest of it is divided in compartments because you have small amounts of space when you need Brownian dynamics individual ion simulation, but a vast amount of intracellular space when you just these ions travel and they can trigger opening channels in the other pocket of space. So that's kind of what is done. If you can kind of overcome this hurdle by using multi-resolution simulation that you can actually simulate over the peak, then you can do some statistics and compare with experiments. For example, one statistics of interest to our collaborators was interpath time. So what is the average time between these spikes? So we can do some statistics, how much, sometimes it's longer this interval, sometimes shorter. But in order to achieve such statistics, such plots, we have to first be able to simulate a system which has few ions, then you have a lot of ions, few ions, a lot of ions, and then if there's a lot of ions, you cannot simulate them all individually. But if there is a few of them, you want to simulate them individually. So this is kind of one type of applications when this could be useful. Say if in your application you can use molecule-based modeling, Brownian dynamics in the whole domain for all times, then there is no need to do this. But this is kind of helping to simulate some system that it's not possible. Another one which application is a collaboration with Radek Erban from University of Maryland. We look at actin modeling. So this is kind of a picture of filopodia, which are fingerless protrusion from cell membranes. So there are some actin filaments, and in these applications, we simulated tip of these growing filopodia using Brownian dynamics, using molecular-based model, and behind it we used just compartment-based modeling, because there's a lot of processes like binding and unbinding happening at the tip. So we wanted to resolve it with the level of individual molecules, but behind it we wanted just to have a quick simulations when these particles are just diffusing or back and forth. So this kind of that type of applications, you basically have some like you need to have a system where you have some small subdomains when you actually feel that you would like to use a detailed model, but when you use such a level of detail in the whole simulations become infeasible. So that's where the multi-resolution modeling becomes, or these type of techniques become very useful. Now this was kind of like an application areas for this, like a coupling from Brownian dynamics to compartment-based modeling. But in both of these applications, we kind of started to think that there are much more information than which is captured by Brownian dynamics. So in both applications, the most detailed models in these papers was Brownian dynamics of points or balls. So proteins were described either by bond-like particles. So if you are interested in exclude the volume effects here, we describe them as balls. But that's a very crude description comparing with what is known about these processes. So in the application areas which we then consider, they include a lot of transport molecules between different parts of the cell. If I summarize this two application areas, macroscopic detail is only required in relatively small regions. And then we apply these methods. But because the molecule structure and functions are known for the sum of these systems, our next question was, can we add these details into multi-resolution simulations? So just to kind of highlight how difficult a task it is for this actin dynamics or the Filopodia simulations, we simulate, our description was just every actin molecule was just a ball. And the information which I like to kind of include there is that it's not a ball, but structure of actins that made from about 2,800 atoms. So an all atom simulations of actin, this would be the underlying model. There's obviously a lot of intermediate models in between, which coarse grain this molecule by saying that some groups of atoms are just particles, in ultra coarse grain, and basically big parts of molecules can be like 4 sides connected by some springs. So there's a question: how can we go from here and start including this level of detail? So that's kind of what brings me to this red arrow, which we are asking the question whether we can add this level of molecular dynamics detail to Brownian dynamic simulations, and the ultimate long-term question is whether we can couple some of these molecular dynamics approaches with particle-based stochastic reaction diffusion methods, like successfully introduce this one to small dyne, whether we can introduce more of it, more detail than actually small dyne or any other reaction diffusion software package provides. So to kind of do this, I have to kind of tell you what is Brownian dynamics. So for my first application, Brownian dynamics was really just simulating of particles as described Brownian motion, and we had this picture. But now in these type of applications, we work on this Brownian dynamics, kind of a more general term. Some people call it Langevin dynamics, when we simulate not just only position, but also velocities of molecules. And we also have to simulate forces. So sometimes in some application, for modern large biomolecules, you don't describe biomolecule just by one point, but by sequence of boards connected by springs. So this is kind of all these type of models I would put to the kind of under this kind of label Brownian dynamics. Now, just to kind of introduce what I mean by molecular dynamics. So the most detailed models which we currently work on are all atom molecular dynamics. And so these are always molecular dynamic simulations in water. So this is a large systems of ordinary differential equation or stochastic differential equation, where we simulate position and velocities of all atoms in the system. So the derivative of position is velocity, and the derivative of velocity is the underlying force. This is just actin molecule. The black is carbon, red is oxygen, blue is nitrogen, yellow is sulfur. But if I want to simulate that, I would have to kind of embed it in water molecules. So there's a lot of water molecules as well. So if I want to do molecular dynamics, there's a lot of choices I have to make: how to use numerical integrators, how to do constraints, how to model bonds, how to simulate systems in the right temperature. And this kind of leads to the typical time step of these simulations are like in the order of femtoseconds, which leaves that molecular dynamic simulations are limited to very, very small domains containing relatively small number of motor molecules, but cells contain a lot of molecules. So there's a lot of like a difference in size.
So Radek, may I pause here and ask one question?
Yes.
So in the previous tutorial session, you showed the one slide where you showed the lots of small particles drive one big particle, and the big particle follows Brownian motion, right? Is that related to this topic?
Yeah, that's very related to this topic, because that's kind of like a type of models. They are theoretical tractable models where you can test some of these ideas, some of these methods before you go to real simulation. So, well, actually, I think I'll mention this model on one of my next slides. So, but before doing that, maybe I should show you some illustrative all atom simulation. So what I will do here, instead of simulating just the whole actin, I'll just simulate a diffusion of ions in water, going to my first application where I have some like a calcium diffusing in water and opening channels. So this is just to give you an idea of what is being done here. So this will be a diffusion of ions in water. So I have to decide how I describe water. And this will be kind of, I will simulate these Newton's equations. So this will be my initial condition. So we can see this is just a box of water molecules. So here you can see red is the oxygen, white is hydrogen. You can see some ions in it. In this visualization, I put one ion, which is kind of a green particle in the middle. Maybe you can let us see it. So a lot of water molecules I have to simulate. So this is my initial condition, and I can just start simulating. So I provide what are the forces between these particles. The typical simulations would look like this. So now in this simulation, what I did, I kind of decreased the sizes of water molecules. So I made them smaller so you can actually see that particle I'm interested in, the ion, the green particle. And you can see that I'm simulating a very large collection of ODEs. So it's kind of chaotic system. And if I would just focus on this green particle, it's just going, it's undergoing some stochastic process. So I kind of understood what that stochastic process is, then I can effectively remove these water molecules and use one of these stochastic reaction diffusion models. So what is my illustrated simulation just show you that all atom MD simulations illustrate the trajectory of ion could be viewed as a stochastic process. So, and the question is whether the stochastic process can be described in terms of few parameters, and that this could help us to couple molecular dynamics with Brownian dynamics. And I would say that was kind of the reason why I introduced this like a more caricature, more models in my tutorial is this mathematician we ask when we do something analytical to get some insights into multi-resolution models in this area. So if you look to these all atom molecular dynamic simulations of ions in the model, they will include both short-range and long-range interactions. So there are interactions, particles are Lennard-Jones interactions and long-range Coulomb forces. But to get some insights into multi-resolution modeling, you can just study analytic tractable molecular dynamics models, which only include one type of interactions. So what I will just show you a model which includes only short-range forces. There's also another one which the long-range forces, but I probably don't have time for it. So let's just concentrate on this short-range forces model, which I already discussed in my tutorial. So that's a kind of Brownian motion of large particle colliding many small particles. So I can maybe just play the video again. So we can, those who attended the tutorial kind of saw this video there. So it's kind of a caricature model when I'm kind of modeling these water molecules now just by these blue point particles, which are going straight until they collide with this big ball, which is modeling a protein. And you can see that in this case, I'm plotting the trajectory of this protein, and it looks like some sort of a Brownian motion. It looks like some sort of a stochastic process. Now, this simulation is kind of a caricature of molecular dynamics simulation, so I can just maybe explain on it what we can do, and then I can just then show some differences from when you work with all atom molecular dynamic simulations. So that was kind of my underlying model. So kind of for this model, what we do, you have to obviously start some good initial conditions. So you assume that the density of light particles is sampled from some distribution, and the velocities are sampled from Maxwellian. So then actually what is nice for these type of theoretical models that one can prove that description in the limit of the mass of these small particles goes to zero. This large particle just satisfies Langevin dynamics, describing in terms of just two parameters, the friction term and the diffusion term. So that's kind of nice about these theoretical models, that one can actually get some theoretical results and prove that what that stochastic process is. Now, this stochastic process can, in the limit, over the end limit also converges to just the Brownian motion, which is used in Brownian dynamics. So what we can do for this theoretical model, we have this like a detailed molecular dynamics model, and we have Brownian dynamics model. Now, detailed model can be described, this can be proven for this type of theoretical model as in terms of Langevin dynamics, and in the limit if I observe system for long times, then this also converges to Brownian dynamics. And these parameters are function, these gamma delta are function of molecular dynamics parameters, where m is the mass of the small particles, capital M is the mass of the big particles, r is the radius of this particle. So I can obtain analytical results, and then you can ask the questions on the Stoy model, what actually means to use models with different level of details of different parts of the computational domain. So basically, you have a part of the domain where you simulate molecular dynamics, when you simulate individual particles, and part of the model when you do Brownian dynamics. And you have to kind of decide what you do when the molecule transfers from this detail model here. That it's subject to collision on one side from the molecular dynamics model, but it's also subject just to stochastic force. So we can obtain some for this particle crossing interface, some analytical formulas for the drift and diffusion coefficient, which are dependent on position and velocity. As long as the particle crosses all the way, then this alpha and beta are equal just minus gamma and gamma square root of 2d. And as long as the particles cross totally to the left, then these alpha and beta are 0. So once you can obtain such a result, you can just start asking the schemes, and when you can use close to the membrane detail model, molecular dynamics, and far away from the site of interest, Brownian dynamics, and you have to use some overlap regions usually where you simulate these systems in parallel. So it's going to be a general idea on this toy model, what is being involved. You have to kind of have establishing some limiting process. Understand that these models are consistent with each other, and then there's an extra thing how to actually design this boundary condition. Now, there are other types of multi-resolution modeling kind of summarized here, that you can obviously not just have an interface when the molecule is crossing, but if you have a biomolecule, you can ask the question, just part of the biomolecule is simulated with detailed model and the rest of it is simulated with less detailed model, or just everything is simulated with and the whole biomolecule is simulated with detailed model, but you want to far away from the biomolecule, use more coarser models. So there are different flavors to multi-resolution modeling in this case. I will skip this other theoretical model just to kind of tell you that what we do in the case of all atom molecular dynamics, then with the kind of the simulation which I showed you. So you have to kind of first find some sort of a equivalent of intermediate approach, which kind of connects molecular dynamics with Brownian dynamics. In this case, it's kind of called generalized Langevin equation, which is kind of some delayed equation. Or you can just equivalently describe it as a larger system of stochastic differential equations. So instead of describing position and velocity, you have additional degrees of freedom. So now in this type of models, which are kind of working at the level of molecular dynamics time step, you kind of don't have water molecules. You describe your important particle, ions, protein by few position, velocity, and a few extra degrees of freedom, which gives you a chance to fit using all atom molecular dynamics simulation to fit these parameters. So that molecular dynamics converges to some suitable generalization of Langevin dynamics, and then this also converges to Brownian motion. So these limiting results can be used to design and analyze hybrid models. And that's what we kind of do, that we run all atom molecular dynamics. We can't prove analytically much for it, but we can use it to parameterize some stochastic coarse-grained model, which are more detailed than Brownian dynamics. And then we can analytically show that these models converge. And because these models are kind of compatible, we can also use it to design some multi-resolution scheme and use in some domain all atom molecular dynamics in outer domain, Brownian dynamics, and intermediate domains you use some coupling regions. So that's kind of what I wanted to kind of mention here. And there's in the last couple of slides, what I discussed, that there are some nice theoretical models which you can use to analyze such a schemes. I discussed Brownian dynamics of ions, and one Brownian dynamic particles, one molecular dynamics particles, but we're also currently working on Brownian dynamics of large biomolecules, that basically we don't coarse grain this as one particle, but as a multiple particles connected by various springs. So kind of give you maybe some idea. I'm going to just move from this paper, when you have basically bead spring model. It's a relatively coarse grain model, but it's of a biomolecule. It's kind of a coarse grain model of DNA, but this is ligand kind of molecule approaching it, and as you approach the molecule, you kind of try to further refine the structure. So basically, you don't use the most coarse-grained model everywhere for the biomolecule, but you use much more detailed model when in the parts of the biomolecule which is of interest. So this is a type of multi-resolution molecule when we are multi-resolving molecules which we use in this area. So we kind of for DNA, but a recent paper when we also work on actin modeling and actin filaments, but try to go from this Brownian dynamics to introduce more biological detail. So if you are interested in this area, so they kind of just before pandemic, we organized some like a Royal Society Kavli meeting, from which there was some special issue on multi-resolution simulation. So it's kind of like it was a snapshot of the work before the pandemic from different research groups. You can find it there. What we are currently trying to bring to it, to bring a more and more detail, like molecular dynamics detail to parts of the biomolecule, to these type of models. So I kind of just summarize my talk. I discuss in this talk multi-resolution modeling, which is all about using models with different levels of detail in different parts of the computational domain. I discuss this red arrow is kind of our success story, because we have papers on the development of mathematical methods and error analysis, but also on application, software implementation, and blue arrow is a work in progress. It's how to introduce more molecular level detail to this Brownian dynamics model through the multi-resolution modeling. There are a lot of unsolved problems, kind of a very active area of research. In my tutorial, I kind of mentioned what kind of on very simple examples what these methodologies are, and it was kind of a short tutorial which is based on this on our new book. So if you're kind of interested in these methods, the videos from our courses. So we have used it as lectures for courses on stochastic modeling for advanced undergraduate and beginning graduate level courses, and for the advanced undergraduate courses we put on YouTube. So you can just find also the videos which we use for teaching undergraduates at our university. It's also fine to just for a self-study on this website. You can also find all the computer codes which are covered, or the methodology which are covered in this textbook. So this kind of brings me to the last slide. So this was a work we've done with a lot of people to acknowledge the funding from funding bodies, most recent ones from Engineering and Physical Sciences Research Council, EPSEX, and from the Royal Society. Thank you very much for your attention.
Thanks so much, Radek. So let's it's a question time. So any question from audiences? Yeah, please unmute yourself and then ask a question.
Can I ask questions?
Please.
I have several questions. Maybe the first one is about when you mentioned DNA structures. So you just, I guess you're considering chemical bonds as springs to connect all the atoms. Am I right?
Well, it depends at which level it's modeled. Yeah. So it's kind of like I mean, what I showed there, that was the most coarsest modeling of polymer. So it's just a bead spring model. So kind of a level that each bead is describing maybe several hundred base pairs. So at that level, it can be described by classical polymer-based models. If you go more detailed model, there are obviously bonds, these all-atom simulations. And they also like have bonds, but you simulate it usually using some constraint algorithms that you fix these bonds and you don't simulate the vibrations of them. So it depends at which level you look at this biomolecular modeling.
What I thought was when we look at that kind of detailed structure of molecules, I think some factors like such as steric hindrance or electrostatic force between atoms would matter as well. I mean, not only for the chemical bonds. So is it okay to like ignore all other chemical things, like rather than chemical bonds?
No, you don't ignore them. There are also like there are bonds, but also interactions. So if you in all atom simulations of biomolecules, there are electrostatic forces. So there are Coulomb interactions between particles which are not connected by bonds. There are also Lennard-Jones interactions. So there are a lot of force terms between parts of the molecule which are not connected directly by bonds, so they are through the chain. So these interactions are all taken into account in the most detailed model, which we are kind of using for parameterizing these coarse grain models. But as you go to the more coarser and coarser models, then you kind of average out a lot of these interactions. So it depends at which level you are looking at. That's kind of a coarse graining. It's a kind of research area in itself that to actually design the structure. This multi-resolution is extra bit to it that you're actually asking to whether you can use the coarse grain model and detail model at the same time in the same dynamic simulation. But for some applications, you can just coarse grain the structure and work with the coarse grain model. You don't need anything multi-resolution.
Yeah, that is right. Thanks. I have one more question about when we couple two regions, I mean molecular-based region and what was it? Compartment-based region. So I remember that the cartoon or animation you showed. So it seems like as time goes by, boundary between compartment-based region and molecular-based region was moving in your animation. So is it easy to implement that kind of simulation? I think I thought it was a little bit complicated to implement. Is it easy? Or
that's a very good question. So in that simulation, maybe I can just put the I think I can just, am I sharing still?
I don't think so. I'm afraid not.
Okay, maybe I can share again.
Yep, please.
So I'm sharing and I'm running a simulation if you see it.
Yeah.
So I kind of stop it now, the simulations. So what is in this simulation is kind of illustrating multiple points. So one point that it illustrates, maybe I can just, it's in a loop, so I'll just stop it initially. It illustrates that you can model this as like a traveling wave problem. So there's some reactions between particles, but also the diffusion. And you can model it classically by partial differential equations. It's kind of like a test problem. So we can, for PDs, you can calculate what is the traveling wave speed, which is the blue, this blue line. You can see that it has some traveling speed. Now, for the compartment-based model, the traveling speed is faster. And for the molecule-based model, it's slower. But it will depend on the parameters, how you choose your compartment size. Now, what we are trying to fit is molecule-based model. And what we are doing, we are doing adaptively. So this is kind of one of the later papers videos, when instead of just postulating when there is the interface is somewhere, the boundary is actually moving as the simulation progresses. So this is when I simulate individual particles, because I need resolving, there are only a few of them. At the beginning of the wave, there are no particles, so it's very discrete. There can be only very few molecules reacting at the front, so that needs to be resolved at the individual particle level. But behind the wave front, there's a lot of them, and compartment-based model is sufficiently detailed. So you do it in a way that you basically calculate the concentration, and based on the concentration passes some threshold, you move the interface, and all these particles which were here will become just one number. So you just add these particles together and move it. So there are some subtleties in this, because you don't want to introduce some bias with it, so you should not do it so often. So the way when you simulate, you have some different time scale, you have some molecular dynamics time step, you can see that things fluctuate here. So there's some like a time, also some like a typical time length when the molecules are produced and degraded, and that's much quicker or smaller time scale than time scale when I'm moving the boundary. So as long as you don't move boundary too often, you don't introduce any bias.
Cool.
Okay. Thank you for the answer.
You find more details in this paper, like these technical details that was taken from.
Thanks.
So questions? Another question, please. Anyone?
May I ask one?
Sure, yeah,
Go
Yuzi,
ahead.
please go ahead.
I have a question about this, the same part, of this connection of the compartment-based model and the molecular based model. So I think the transition from molecular-based to compartment-based model occurs when, the number of the particles exceeds certain value. So